Behind the closed doors of the Vatican's secret laboratories, things are happening that are not usually spoken about openly. Genetic analysis of sacred relics reveals data that could overturn our understanding of Christian history. DNA from the Shroud of Turin, blood on the nails of the Crucifixion, the remains of saints. All of this is being studied, and the results are carefully guarded from the public. What is this data hiding? Why are insiders risking their careers to tell the truth? Today, we will dive into a world where science meets faith, and discoveries raise more questions than answers. If you are interested in the secrets that official history prefers to keep quiet, subscribe to the channel and like this video. And in the comments, write your opinion. Does science have the right to study sacred relics? Or are there boundaries that should not be crossed? This is one of the most controversial issues of our time, and your opinion is important. In 1988, a group of scientists obtained permission to do what seemed impossible: take a sample of fabric from the Shroud of Turin for radiocarbon analysis. This artifact, kept in the Italian city of Turin, has been considered the burial shroud of Jesus Christ for centuries. The 4-meter-long, 36-centimeter-wide cloth bears the imprint of a crucified man with characteristic wounds from a crown of thorns, nails, and a spear. For centuries, millions of pilgrims have bowed before this cloth, believing that they are touching the traces of a divine presence. The results of the first scientific study were to either confirm the antiquity of the relic or debunk centuries of belief. 3 independent laboratories in Oxford, Arizona, and Zurich received tiny fragments of fabric measuring only a few square centimeters. Each laboratory worked independently, unaware of the results of the others. When the data was compared, the scientific world held its breath in anticipation. The dating indicated a period between 1260 and 1390, the Middle Ages, the era of the Crusades, and the heyday of the relic trade. For many believers, this came as a shock. But the controversy did not end there. Critics of the methodology pointed to possible contamination of the samples, the impact of the fire of 1532, and the peculiarities of the weaving of the threads. However, few people knew that, in parallel with the dating, other studies were being conducted, attempts to extract and analyze biological material from the fabric. Stains that looked like blood caught the attention of geneticists. If it really was blood, whose was it? Was it possible to extract DNA from a sample that was almost 2,000 years old or 700 years old, according to radiocarbon dating? The technology of the late 1980s was not yet advanced enough for a full genetic analysis of ancient samples, but attempts were made. The samples were preserved and placed in special storage facilities. With the development of DNA sequencing methods in the 2000s, these samples were revisited. In 2015, researchers from the University of Padua published data showing that DNA of plant and human origin had been found on the shroud. The human genetic traces belonged to many people from different geographical regions, from the Middle East to Western Europe, and even India. This was explained by the fact that thousands of pilgrims had touched the relic over the centuries of its existence. But the researchers stumbled upon something strange. Some genetic markers did not match known populations. The samples contained sequences that did not fit into standard databases. This raised the question: was this the result of ancient DNA degradation, or something else? The Church has always been cautious about scientific research on the shroud, never officially recognizing its authenticity, but also never refuting it. The Vatican called the artifact an icon rather than a relic, leaving the question of faith to the discretion of each Christian. However, behind closed doors, completely different conversations were taking place. Access to the results of genetic research was strictly limited, and the data was not published in open scientific journals. Only fragmentary intellectual leaked through insiders, scientists who participated in the projects but later decided to break their vow of silence. They talked about strange coincidences in the genetic code, about sequences that should not have been present in a tissue sample from 1st century Palestine. Some even claimed that part of the genetic data had been deliberately removed from the official reports. What frightened the researchers so much? Why were the results, which could shed light on the greatest mystery of Christianity, banned? Secret laboratories do not only exist in conspiracy theories. They are real and located in the heart of the Vatican. Officially, the Catholic Church does not conduct genetic research on relics, but documents leaked to the press in 2010 tell a different story. In the basement of the Vatican museums, in buildings where ordinary tourists are not allowed, there are scientific laboratories equipped with the most modern equipment. Not only Italian specialists work here, but also invited geneticists from leading universities around the world, who sign non-disclosure agreements for a period of 25 years. What are they researching behind these closed doors? The Vatican has one of the largest collections of Christian relics in the world, from fragments of the Cross of the Lord to the relics of apostles and saints. Many of these artifacts have been stored for centuries without proper scientific cataloging, their authenticity based solely on church tradition and documents that could themselves be forgeries. With the development of genetic technology in the late 20th century, the Church had the opportunity to verify the authenticity of its treasures using scientific methods without making it public. The project began in 1993 under Pope John Paul II, who had always been interested in science and saw no contradiction between faith and knowledge. A closed commission was created under the code name Veritas, Truth. It consisted of cardinals, theologians, historians, and scientists. Their task was to determine which of the relics stored in the Vatican deserved to be considered authentic and which were pious forgeries from the Middle Ages. This required comprehensive research, radiocarbon analysis, the study of textile fibers, chemical analysis of dyes, and most importantly, genetic testing of biological remains. However, it quickly became clear that some of the results were better kept secret. The first object of study was a relic known as the Holy Blood, an ampule containing a substance that supposedly contained the blood of Christ, collected by Joseph of Arimathea. Analysis showed that the liquid did indeed contain hemoglobin, but dating indicated the early Middle Ages, approximately the 7th or 8th century. The genetic material belonged to a man of Mediterranean origin. The result was ambiguous. The relic turned out to be ancient, but not as ancient as tradition claimed. The commission decided not to publish this data, limiting itself to an internal report. The next stage was the study of the relics of saints kept in Roman basilicas. The remains of the Apostle Peter, discovered during excavations under St. Peter's Cathedral in the 1940s, were long considered an indisputable find. Genetic analysis of the bones, conducted in 2003, showed that they belonged to a man between the ages of 60 and 70, who lived in the 1st century AD. Isotope analysis showed that the place of origin coincided with the region of Galilee. According to the Gospels, this is where Peter came from. It seemed like a triumph, but the researchers faced a problem: how to prove that these bones belonged to the Apostle Peter and not to any other inhabitant of Galilee at that time. For final identification, reference DNA from relatives was required, which of course did not exist. Then the idea arose to compare the genetic material with other relics associated with Christ's close circle. An unprecedented project began to collect genetic data from all available Christian artifacts containing biological material. The geography of the research expanded far beyond the Vatican. Laboratories requested samples from churches around the world. Under various pretexts, most often for restoration or conservation, tiny fragments of relics were sent to Rome. Not all churches agreed to provide samples. Orthodox churches in Russia and Greece refused to participate in what they called desecration of holy sites. However, Western Catholic institutions were more accommodating. By 2007, a unique database containing genetic information on dozens of relics had been compiled. And that's when the really strange discoveries began, forcing the project's leadership to tighten security. What did scientists find in these ancient samples? Genetic analysis of ancient remains is an art that balances on the edge of what is possible. DNA deteriorates over time, especially in less than ideal conditions. Temperature fluctuations, humidity, microbial contamination, ultraviolet radiation. All of these factors transform once intact molecules of heredity into fragmented chains that are difficult and sometimes impossible to read. However, the technologies of the last 2 decades have revolutionized the field of paleogenetics. What seemed like science fiction in the 1990s has become routine in the 2000s. The polymerase chain reaction, or PCR method, has made it possible to amplify, multiply, even the smallest fragments of DNA from ancient samples. If at least one intact molecule remains in the sample, it can be multiplied to a quantity sufficient for analysis. The next breakthrough was next-generation sequencing, a technology that allows millions of short DNA fragments to be read simultaneously and then assembled into a single sequence, like a mosaic of thousands of pieces. It was these methods that were applied to relics in secret Vatican laboratories. But even the most advanced technologies face fundamental limitations. The main problem is contamination. The contamination of modern DNA. Every person who has ever touched a relic has left their genetic traces on it: skin cells, hair, saliva. Archaeologists, museum curators, restorers, pilgrims, all of them have become unwitting contaminators. Ancient DNA can be distinguished from modern DNA by characteristic damage. In ancient samples, cytosine often turns into uracil, creating specific error patterns. These timestamps allow researchers to filter out modern contamination and focus on genuinely ancient material. However, what if ancient DNA itself contains unexpected elements? In 2009, a group of researchers working with samples from the Vatican's reliquaries discovered unusual mitochondrial DNA in one of the samples of the relics of Saint Anne, the mother of the Virgin Mary. Mitochondria are the energy powerhouses of cells, which have their own small genome that is passed down exclusively through the maternal line. Studying mitochondrial DNA allows us to trace maternal lines over thousands of years and determine a person's geographical origin. The sample was indeed ancient. All markers indicated an age of about 2,000 years, but the mitochondrial haplogroup did not match any of the known populations of the Middle East from that period. It was close to the haplogroups characteristic of North Africa, but had unique mutations that were not found in any modern or ancient database. The result was so unusual that the researchers conducted a reanalysis, taking new samples from another part of the remains. The result was confirmed. Either this was not Saint Anne, or her genetic origin was completely different from what tradition had assumed. The Veritas Commission faced a dilemma: to publish the results and cast doubt on the authenticity of the relic, or to remain silent. It was decided to conduct additional research on other relics associated with the family of Christ before drawing any conclusions. At the same time, work was carried out on the Shroud of Turin. Despite radiocarbon dating in the 1980s, which pointed to the Middle Ages, many scientists continued to believe that the analysis had been carried out on a section of fabric that had been restored or contaminated. In 2012, a new group of researchers obtained permission to extract biological material from blood-like stains. Using non-invasive methods, they collected microparticles from the surface of the fabric, using special adhesive tape. Genetic analysis revealed the presence of human DNA, but it was highly fragmented and mixed with the DNA of many other people. Nevertheless, it was possible to isolate several sequences that were highly likely to belong to a single male individual. The Y chromosome, which is passed from father to son, showed a haplogroup characteristic of the Middle East. But upon closer analysis, they found sections that did not fit into standard reference databases. Some non-coding regions contained repetitive sequences of unusual length and structure. One of the geneticists involved in the project later reported anonymously in an interview with a scientific journal that he had encountered genetic markers that he could not classify. According to him, this could be a consequence of the extreme age of the sample and DNA degradation. But it could also indicate something unusual in the genetic structure itself. His comments were cautious. He avoided sensational statements but hinted that the full data had not been published. Sequencing technologies developed rapidly, and by the mid-2000s, it became possible to read the entire human genome from ancient remains, provided the material was sufficiently well preserved. Vatican researchers applied this technology to several of the best-preserved samples of saints' relics. They hoped to find genetic links between relics that, according to tradition, belonged to relatives and thus confirm their authenticity. But the results were unexpected and even disturbing. Why did the whole genome sequencing data cause such concern among researchers? The history of the relics of Saint Longinus the Centurion is shrouded in legends no less than the history of the character himself. According to Christian tradition, Longinus was a Roman legionnaire who pierced the ribs of the crucified Jesus with a spear to confirm his death. Drops of blood and water spurted from the wound, splashed into the soldier's eyes, and miraculously healed him of his blindness. Shocked by what had happened, Longinus believed in Christ, left the service, and became a preacher, for which he was subsequently executed. According to legend, his remains were found in Cappadocia in the 6th century, and later transferred to various cities in Europe. Parts of Longinus's relics are kept in several places: the Roman Basilica of Santa Maria in Trastevere, the Monastery of Mantua, and the Cathedral of Landshut in Germany. In 2011, the Vatican decided to conduct a comparative genetic analysis of samples from all these locations to determine whether they all belonged to the same person. The results were contradictory. Samples from Rome and Mantua did indeed show genetic kinship. They belonged to a single male individual who lived around the 1st century AD. Isotopic analysis of tooth enamel indicated a childhood spent in the Mediterranean region, which corresponded to the legend of a Roman soldier. Everything looked convincing, and the researchers were already preparing to announce the confirmation of the relic's authenticity. But the analysis of the Y chromosome brought a surprise. The haplogroup was not what they expected to see in a Roman legionnaire. 1st century Roman soldiers were recruited from all corners of the empire, but the vast majority belonged to European or Mediterranean genetic lines. The haplogroup found in Longinus's remains was characteristic of the population of the Levant, a territory that includes modern-day Israel, Lebanon, Syria, and Jordan. In other words, genetically, this man was not Roman, but a local resident of the region. Is it possible that the Roman legionnaire was originally from Judea? Theoretically, yes. After the suppression of the Jewish uprising in 70 AD, the Romans did recruit soldiers from the local population, but the legend of Longinus referred to the events of 33 AD, when Christ was crucified during the reign of Pontius Pilate. At that time, local residents rarely served in the Roman legions, especially as executioners in a religiously tense region such as Jerusalem. This contradiction led researchers to question either the authenticity of the relic or the veracity of the legend itself. But the real shock was yet to come. Detailed genome sequencing revealed unusual sections on the 3rd chromosome. There was a cluster of genes associated with the development of the eyes and visual system. And in this region, there were mutations that modern genetics associates with hereditary forms of blindness or serious visual impairments. The probability of a random coincidence was negligible. It turned out that the person whose remains were being studied did indeed have a genetic predisposition to blindness or poor vision. Legend has it that Longinus was blind until his miraculous healing. If this was a coincidence, it was remarkable. If not, how should we interpret the connection between genetic data and religious tradition recorded centuries after the events? Further analysis revealed another oddity. The genome showed traces of a recent, by population genetics standards, mixing of different genetic components. It looked as if this person's ancestors belonged to two significantly different populations that had mixed just a few generations before his birth. This could have happened if one of the parents was from a completely different region, perhaps North Africa, or even more distant lands. This was unusual for the 1st century, though not impossible. The Roman Empire provided a certain degree of population mobility. One of the geneticists who worked with Longinus's samples later said in a confidential conversation that the team encountered DNA segments that they could not classify accurately. They did not match any known ancient population, although they were close to Levantine groups. This could be explained simply by a lack of data. The database of ancient genomes from the Middle East of the 1st century is very limited. But there could be another explanation, which they preferred not to talk about openly. The Vatican Commission decided to classify the full data of the genetic analysis of St. Longinus's relics. The official report contained only confirmation of the antiquity of the remains and their belonging to a man of Mediterranean origin. Not a word was said about genetic abnormalities, mutations associated with blindness, or the unusual mixing of populations. The samples were returned to the reliquary, and access to them was closed for an indefinite period. The researchers signed additional non-disclosure agreements. But information has a way of leaking out, especially when it comes to something that could overturn established ideas. Insiders began to share fragments of data, and discussions appeared on specialized forums of genetic scientists. Rumors began to circulate about problematic samples, relics whose analysis results did not fit within the expected framework. The relics of Longinus were the first, but far from the last, case where genetics raised more questions than it answered. What else did the results of research on other sacred artifacts reveal? Blood on ancient artifacts has always been of particular interest. It is the only biological material that could have been preserved on religious objects that are not directly remains. The spear of destiny, the nails of the crucifixion, fragments of the crown of thorns. All these relics, according to legend, came into contact with the blood of Christ. If this is true, genetic analysis could answer a question that has preoccupied humanity for 2,000 years. Who was Jesus from a biological point of view? Longinus's spear, also known as the Spear of Destiny, is kept in the Hofburg Treasury in Vienna. It is a 1st century Roman spearhead, embedded in a late medieval spear. According to tradition, it was with this spear that a Roman soldier pierced Christ's side on the cross. There are indeed dark spots on the tip that could be oxidized blood. In 2014, after lengthy negotiations, the Viennese authorities allowed microscopic samples to be taken from these spots for analysis. The work was carried out in strict secrecy. The results of radiocarbon dating of the spots were ambiguous. The organic material was ancient, but it was not possible to determine the exact date due to the small size of the sample and possible contamination. Genetic analysis showed the presence of male human DNA, but it was so fragmented and degraded that it was not possible to obtain a complete sequence. Only a few short sections of mitochondrial DNA and the Y chromosome could be identified. These fragments indicated a Middle Eastern origin, which was consistent with the historical context. But was this the genetic material of Jesus Christ, the soldier who held the spear, or someone else whose blood got on the artifact at any point during its 2,000 years of existence? It was impossible to prove this scientifically. A similar story occurred with the nails of the crucifixion. Dozens of authentic nails with which Christ was allegedly nailed to the cross are scattered throughout the Christian world. The most famous ones are kept in Rome, Venice, Milan, Prague, and Jerusalem. In 2016, the Vatican organized a secret program to collect samples from the most revered of these relics. Traces of organic material, which could be human tissue or blood, were found on some of the nails. Genetic analysis revealed human DNA, but in most cases it belonged to several different individuals, probably people who had touched the relics over the centuries. However, 2 nails, from Rome and Venice, were found to have genetic sequences that most likely belonged to the same man of Middle Eastern origin. Moreover, this man's mitochondrial DNA was identical to that found on a fragment of the crown of thorns kept at Notre Dame de Paris before the fire of 2019. The coincidence was striking. 3 different relics, stored in different cities, contained the genetic material of one person. This either confirmed their authenticity or pointed to the medieval practice of replicating relics, when one original blood sample was used to create several authentic artifacts. The researchers leaned toward the second explanation, but could not rule out the first. Then the idea arose to compare these sequences with data obtained from the Shroud of Turin and the relics of St. Longinus. If a genetic match could be found between different relics, it would create a network of cross-confirmations pointing to a real historical event. The result was shocking. The mitochondrial DNA from the nails and the crown of thorns did not match either the shroud or the relics of Longinus. Moreover, it did not match any other samples from the Vatican database. This meant one of 2 things. Either we were dealing with truly unique genetic material unrelated to any other known relics, or it was the blood of a medieval man used to create fake artifacts. Y chromosome analysis added another layer of complexity. The haplogroup indicated Levantine origin, but contained mutations that were rare even for that region. Statistical analysis showed that this combination of mutations occurs in approximately 1 in 10,000 people in modern Middle Eastern populations. In ancient times, when genetic diversity was even greater, this made the carrier of such a Y chromosome almost unique. Some researchers interpreted this as a sign of authenticity. Indeed, if medieval forgers had used someone's blood, it would have been genetically typical of medieval Europeans, not ancient Levantines with rare genetic markers. But critics objected. In the Middle Ages, Crusaders brought back not only relics from the Holy Land, but also people, slaves, mercenaries, merchants. The blood of any of them could have been used to create authentic artifacts. There was only one way to resolve this dispute: find direct genetic relatives and compare DNA. But where to look for living descendants of a person who lived in the 1st century? That was when the most ambitious and controversial idea in the history of religious genetics was born: the Bethlehem Project. What were the goals of this secret project, and what conclusions did its participants reach? The Bethlehem Project was launched by the Vatican in 2017 under the guise of an archaeological and anthropological program to study the population of ancient Judea. Officially, its goal was to create a database of ancient genomes from the Holy Land to better understand the migrations and genetic history of the region. But the real task was much more ambitious: to find living people who could be distant genetic relatives of those whose DNA was found on sacred relics. The project was based on simple genetic logic. The Y chromosome is passed from father to son with virtually no changes over thousands of years. Mitochondrial DNA is also passed from mother to children with almost no changes. If there are people in modern Middle Eastern populations with a Y chromosome or mitochondrial haplogroup identical or very close to that found on the relics, this could indicate genetic continuity dating back to the 1st century. Geneticists from universities in Israel, Lebanon, Jordan, and the Palestinian territories were recruited to carry out the project. They were officially told that data was being collected for a population genetic study funded by an international scientific foundation. In reality, the project was coordinated by Vatican officials, and the samples were secretly compared with genetic data obtained from sacred artifacts. Over 3 years, more than 20,000 DNA samples were collected from volunteers living in Israel, Palestine, Lebanon, Syria, and Jordan. Particular attention was paid to old religious communities: Aramaic Christians, Samaritans, Druze and Palestinian Christians from ancient families. These groups had lived in the same territories for centuries and were able to preserve genetic lines dating back to antiquity. Data analysis began in 2019, almost immediately similarities were discovered. Several Palestinian Christians from the Bethlehem and Nazareth areas were found to have Y chromosome haplotypes that were highly likely to have a common ancestor with the haplotype found on the nails of the Crucifixion and the crown of thorns. Statistical calculations showed that the common ancestor lived about 2 millennia ago, give or take a few centuries. It was a sensation, but it could not be made public. If modern Palestinians are genetically linked to the relics of the Crucifixion, what does that mean? That they are descendants of Christ's family? Or simply representatives of the same population to which the executed man belonged? It was impossible to scientifically prove that the DNA on the relics belonged to Jesus himself. This would have required samples from his parents or children, whom, according to Christian tradition, he did not have. Moreover, other coincidences were discovered. A group of Lebanese Maronite Christians was found to have a mitochondrial haplogroup identical to that found on the relics attributed to St. Anne. Israeli Samaritans, an ancient religious group that considers itself to be direct descendants of the tribes of Israel, were found to have Y chromosome lines similar to those found on other first-century relics. A picture gradually emerged. The genetic lines associated with sacred artifacts have not disappeared. They continue to exist in modern Middle Eastern populations, especially among those groups that have maintained religious and cultural isolation. But then the researchers encountered a problem. Upon closer analysis, it turned out that some of these genetic lines contain sections that do not fit into the standard evolutionary model of the human genome. These were non-coding regions, sections of DNA that do not carry information about proteins but can influence gene regulation. In these regions, repetitive sequences of unusual structure and length were found. One of the geneticists involved in the Bethlehem project, who later gave an anonymous interview to a scientific publication, compared these sequences to genetic palimpsests, as if layers of information had been written into the genome, which were then partially erased, but traces remained. He did not claim that this was a sign of something supernatural, but noted that standard evolutionary models do not explain the origin of these structures. Could this be the result of ancient genetic events, for example, mixing with some unknown extinct population, similar to how modern humans carry Neanderthal and Denisovan genes? Theoretically, yes, but analysis showed that these sequences were unlike either Neanderthal or Denisovan ones. They were unique. Another hypothesis was that this was simply the result of genetic drift and random mutations that had accumulated in isolated populations in the Middle East. But then, why were such sequences concentrated precisely in those genetic lines that were associated with first-century relics? The coincidence seemed too implausible. The Bethlehem Project leadership faced a dilemma. Publishing the full data could provoke an uncontrollable reaction, ranging from theological disputes to political conflicts in the Middle East. If modern Palestinians are the genetic descendants of Christ's family, how would this affect the Israeli-Palestinian conflict? Who would get the rights to the relics? How would different Christian denominations react? In 2020, as the Bethlehem Project was nearing the final stage of analysis, information was leaked. An unknown employee of one of the laboratories passed on to journalists partial data on genetic matches between the relics and modern Middle Eastern populations. The story appeared in several scientific blogs and specialized publications, but did not receive widespread attention. Most readers dismissed it as yet another conspiracy theory. The Vatican immediately issued a denial, stating that no genetic research had been conducted on the sacred relics, and that the aforementioned Bethlehem Project was a routine academic study of population genetics. The universities involved in collecting the samples confirmed this version. All publications were quickly removed under threat of legal action. However, closed discussions began in the scientific community. Some geneticists not involved in the project requested access to the data for independent verification, but were denied on the grounds of ethical restrictions and the need to protect the confidentiality of the study participants.
Meanwhile, a rift developed within the project team. Some scientists believed that the results should be published in full, regardless of the possible consequences. Science cannot serve political or religious interests. Others insisted on caution, pointing to the unpredictability of public reaction and the potential danger to the carriers of these rare genetic lines themselves. What if radical groups decide that these people have some kind of special holy blood and begin to persecute them, or, conversely, glorify them? History knows many examples of genetic information being used for dangerous ideological purposes. The decisive meeting took place in the Vatican in January 2021. It was attended by cardinals from the Congregation for the Doctrine of the Faith, representatives of the Pontifical Academy of Sciences, and leaders of the Bethlehem Project. The discussion lasted 3 days. In the end, a compromise was reached. Only the data concerning the population genetics of the region would be published, without mentioning the connection to the relics. All samples taken directly from the artifacts were declared insufficient for publication. The genetic data of carriers of rare haplotypes is anonymized to such an extent that it becomes virtually impossible to establish their identity. The project is officially closed with the statement that the research objectives have been achieved. But the most significant discoveries remained behind closed doors. Among them was one that was not even mentioned in the secret reports available to most project participants. Only a small group of 5 people knew that when comparing the genomes of modern carriers of rare haplotypes with ancient samples, a correlation was found not only in the main genetic markers, but also in those very anomalous non-coding sequences. In other words, the strange genetic structures found on first-century relics were also present in their presumed modern descendants. This meant that we were not dealing with an artifact of ancient DNA degradation or the result of sample contamination, but with a real inherited genetic trait that had been passed down from generation to generation for 2 millennia. The functional significance of these sequences remained unclear. They did not affect the coding regions of genes, did not cause disease, and were not associated with any measurable phenotypic traits. But their stable inheritance and uniqueness raised the question: was this a random genetic feature of a small population, or something else? One member of the small circle, a molecular biologist with 30 years of experience, put forward a hypothesis that seemed too speculative to the others. Perhaps these sequences are remnants of ancient genetic information that has been lost in most human populations, but has been preserved in a few isolated lines in the Middle East. He compared it to the genetic memory of a population whose function is still unknown to us. This idea was not developed further. It was considered too speculative to be included even in a closed report. The materials of the Bethlehem Project were placed in a special Vatican archive with restricted access, which allows them to be viewed only with the permission of the Pope. Digital copies of the data are stored on secure servers without an internet connection. All project participants received generous grants to continue their research in other areas, on the condition that they never return to the topic of relic genetics. Most agreed, but there are always those who cannot remain silent when they believe that humanity has a right to know the truth. It was from these sources that fragments of information began to leak out, forming a picture of what was really happening behind the closed doors of the Vatican laboratories. However, this was only the beginning of a story that was becoming increasingly confusing and contradictory. The next stage was the study not so much of the relics themselves as of the contradictions in their dating and origin, which cast doubt on the official versions of their history. Contradictions in the dating of relics have haunted researchers since the very beginning of the application of scientific methods to sacred artifacts. Radiocarbon analysis of the Shroud of Turin in 1988 pointed to the Middle Ages, but supporters of its authenticity pointed to dozens of factors that could have skewed the result. The fire of 1532, during which the fabric was exposed to high temperatures, could have added newer carbon. Centuries of pilgrims touching the shroud left organic contaminants. Restoration attempts in different eras could have introduced modern fibers. Each of these factors could theoretically shift the date by centuries. But the problem was deeper. Different dating methods gave different results. When, in 2013, a group of Italian scientists applied an alternative method based on the analysis of the mechanical properties of cellulose fibers and their degradation, they obtained a date indicating a period between the 1st century BC and the 1st century AD. The difference with the radiocarbon method was more than 1,000 years. Was this an error in one of the methods, or were the two methods measuring different things? Could the fabric have been ancient, but contaminated with medieval material to such an extent that the radiocarbon analysis gave an erroneous result? Similar contradictions arose when dating the relics of saints. Remains that, according to church tradition and historical documents, should have belonged to the early centuries of Christianity, sometimes turned out to be several centuries younger when analyzed by radiocarbon dating. Explanations ranged from errors in historical records to the medieval practice of replacing lost relics with new remains, which were attributed the identity of the former. The Church has repeatedly faced the fact that relics venerated for centuries turned out to be the remains of people from a completely different era. But there were also opposite cases. Relics that historians considered to be medieval forgeries turned out to be genuinely ancient upon analysis. This was the case with a fragment of a wooden cross from the monastery of St. Catherine on Mount Sinai. Tradition attributed this fragment to the cross found by St. Helena in the 4th century. But historians were skeptical. Too many particles of the true cross are scattered throughout the Christian world. And if you put them all together, you would end up with not a cross, but a whole forest. However, radiocarbon dating of the Sinai fragment indicated the 1st century AD, plus or minus 50 years. Analysis of the type of wood showed that it was olive, which grows in the region of Judea. Dendrochronological research, a method of dating by annual rings, confirmed the result. The relic turned out to be genuinely ancient, although it was still impossible to prove that this particular piece of wood was part of the cross of the Crucifixion. The strangest contradictions arose during a comprehensive analysis of several relics, which, according to legend, were associated with the same event. In 2018, a group of researchers gained access to 3 artifacts stored in different European cathedrals. A fragment of fabric believed to be part of Christ's burial shroud, a sample of wood from the cross, and a piece of stone allegedly from the Holy Sepulchre. All 3 were independently dated using several methods. The results were shocking. The fabric dated back to the 1st century, the wood to the 1st or 2nd century, but the stone was much older, approximately 200 BC. What did this mean? Either the stone was not related to Christ's burial, or the tomb in which he was buried had been carved out of the rock 2 centuries before this event and was reused, which, however, corresponds to the Gospel description. Joseph of Arimathea provided his own, already prepared tomb, but genetic analysis added even more confusion. Traces of human DNA were found on the fabric and on the wooden fragment as well, although in much smaller quantities. Researchers expected that if all 3 artifacts were related to the same event, the genetic traces would match. But the mitochondrial DNA from the fabric did not match the DNA found on the wood. Moreover, no biological material of ancient origin was found on the stone at all, only modern contamination from tourists and clergy. There were several possible explanations. First, the artifacts are not related to each other and are a set of disparate objects to which a common legend has been attributed. Second, they do indeed come from the same place and time, but the biological material on them belongs to different people. For example, those who participated in the burial. Third, over 2 millennia, the relics have been subjected to so much manipulation, replacement, and restoration that it is no longer possible to establish their true origin. Researchers leaned toward the third explanation, but could not completely rule out the other options. The case of the blood on the cross was particularly intriguing. Genetic analysis showed that it was the blood of a man of Middle Eastern origin who lived in the 1st or 2nd century. But the Y chromosome haplogroup was different from that found on the nails of the Crucifixion and the crown of thorns. If all these relics are connected with the crucifixion of Christ, why do the genetic traces not match? Either the blood of different people remained on different artifacts, which is quite possible, considering that the Romans used crosses repeatedly to execute different criminals. Or not all of these artifacts are authentic, and only some of them are actually connected with historical events. The Vatican preferred not to comment on these contradictions publicly, limiting itself to a general statement that the authenticity of relics is a matter of faith, not science. But within church structures, the contradictory data sparked serious discussions that continue today. The ethical dilemmas associated with scientific research into religious relics go far beyond a simple conflict between faith and reason. They raise fundamental questions. Does science have the right to intrude into the sacred sphere? Who owns relics? The church that has preserved them for centuries, the believers who revere them, or all of humanity, which has a right to know the historical truth? Is it possible to apply destructive methods of analysis to artifacts that for millions of people are not just historical objects, but a channel of communication with the divine? To perform genetic analysis, it is necessary to physically take a sample, albeit microscopic, but still a sample. This means partial destruction of the relic. For believers, this can be perceived as desecration. The Orthodox Church traditionally takes a tough stance. The relics of saints should not be subjected to scientific research, because holiness cannot be measured by instruments. The Catholic Church is more flexible, but also cautious. Permission to take samples is given extremely rarely and only after lengthy internal discussions. Protestant denominations, which are generally less attached to the veneration of material relics, take a more relaxed view of the issue. But even among them, there are conservative currents that see genetic analysis of relics as an attempt to reduce the divine to the level of biology. One of the central ethical questions is what to do with the results if they contradict religious tradition. In 2010, Russian scientists received permission to conduct research on the relics of Saint Seraphim of Sarov, one of the most revered Orthodox saints. The analysis showed that the remains belonged to a man who lived in the 18th to 19th centuries, which corresponded to the saint's biography. But genetic analysis revealed signs of several hereditary diseases that were never mentioned in hagiographic tradition. They did not fit the image of the saint as a spiritual athlete and miracle worker. Should this data be published? The Church was against it. Scientists insisted on the completeness of scientific information. No compromise was ever reached. Some of the data remained classified. An even more heated debate surrounds the commercialization of genetic research on relics. Several private laboratories offer genetic analysis services for religious artifacts for significant sums of money. Churches in poorer countries that own relics sometimes agree to such research in the hope that scientific confirmation of authenticity will attract more pilgrims and donations. This turns sacred remains into a commodity, which contradicts the religious ethics of all faiths, but it happens in practice. There is also a political dimension to the problem. When, in 2016, genetic analysis of relics from a Bulgarian monastery showed that the remains belonged to a woman, although they were revered as the relics of Saint John of Rila, one of the patron saints of Bulgaria, it caused not only a religious scandal but also a national one. Saint John is a symbol of Bulgarian identity, and the news that the relic might be fake was seen as a blow to national pride. The government and the Patriarchate issued a joint statement questioning the research methodology. The research team was subjected to pressure and threats. The results of the study were never fully published. Cases like this make scientists wonder: is the truth worth the social upheaval that may follow its disclosure? Another ethical aspect is consent to research. When it comes to modern humans, any genetic research requires their informed consent. But who can give consent for research on the DNA of a person who died 2,000 years ago? Their descendants? But how can they be identified? The religious community that reveres them as a saint, the state in whose territory the remains are located, the church that is their custodian? Each of these parties has its own interests, and they often conflict. A particularly difficult situation arises when genetic analysis reveals family ties. The Bethlehem Project faced this problem head-on. If modern people are genetically related to ancient relics, do they have any rights to these artifacts? Should they even know about their genetic relationship to saints or biblical figures? Most of the project participants leaned toward no. This information could radically change people's lives, and not necessarily for the better. Imagine being told that your mitochondrial DNA is identical to that found on relics attributed to the Virgin Mary. How would that change your life? Would you become the object of pilgrimage, persecution by sects, or scientific study? Would you feel a special responsibility? Or, on the contrary, the burden of this knowledge? Did scientists have the right to hide this information from the carriers of unique genetic lines themselves? International bioethics committees have not yet developed clear recommendations on this issue. Existing protocols were developed for modern medical research, not for the genetics of ancient relics. Each case is considered individually. Decisions are made ad hoc, without a unified methodological basis. This creates room for manipulation. Some studies are blocked on ethical grounds, while others are conducted in secret without any ethical oversight. The Vatican has developed internal ethical standards for relic research, but these remain confidential and are not subject to external audit. This has drawn criticism from the scientific community. How can the results of research be trusted if the process is not transparent? But the Church counters, how can we trust scientists who do not understand the spiritual dimension of what they are researching? This conflict is far from being resolved, and while it continues, many potentially important studies are being postponed or conducted in secrecy. This further exacerbates the problem of trust and provokes conspiracy theories about the concealment of the truth. Leaks from closed projects on the genetic analysis of relics began almost immediately after the first results were obtained. In 2012, an anonymous user under the pseudonym Veritas33 began publishing fragments of data on a specialized genetics forum, which, according to him, were obtained during Vatican research. The first publications were met with skepticism. The claims of genetic abnormalities in the relic samples seemed too incredible. But when Veritas33 published specific genetic sequences with precise coordinates in the genome, some experts began to verify the data. It turned out that the sequences were real. They did not match any known genome, but technically looked authentic, with no signs of falsification or random noise. It was not possible to establish who Veritas33 was. He used anonymous networks and encryption. In 2015, his activities suddenly ceased, with his last message stating, "I have been asked to remain silent." Perhaps that is right. Some knowledge is dangerous. But by that time, the information had already spread across dozens of scientific blogs and conspiracy websites. Another source of leaks was insiders, scientists who participated in the research but were dissatisfied with the secrecy regime. One of them, a molecular biologist from the University of Padua, gave an interview to an Italian scientific journal in 2017 on condition of complete anonymity. He spoke about problematic samples, relics whose genetic analysis yielded results that did not fit into standard evolutionary models. According to him, some samples contained genetic sequences that looked like insertions, sections of DNA that did not appear as a result of natural mutations, but seem to have been added from outside. He emphasized that this does not necessarily mean anything supernatural. There are many sections of the human genome whose origin is not entirely clear, including the remains of ancient viruses embedded in our DNA millions of years ago. But the pattern of these sequences in the relic samples was unusual, and the team could not find a convincing explanation for them. When the interview was published, the journal was inundated with criticism. The Vatican issued an official denial, stating that the research described had never been conducted and that the source was either mistaken or deliberately misinforming the public. The University of Padua distanced itself from the publication, noting that it could not confirm the identity of the source or the veracity of his claims. The journal was forced to publish an apology and remove the article from its online version, but copies had already spread across the internet. The third category of leaks came from technical staff, lab assistants, system administrators, archivists. These people did not always understand the scientific significance of the data they were working with, but they could provide documentary evidence, photographs of documents, screenshots of databases, internal correspondence. In 2019, a photograph of an internal report by the Vatican's Veritas Commission, marked strictly confidential, was leaked online. The low-quality photograph showed a table with the names of relics in the left column and genetic haplogroups in the right. Several lines were highlighted in red with the Italian note, "Anomalie da approfondire," anomalies requiring further study. The authenticity of the document could not be verified. It could have been either a real leak or a clever forgery. The Vatican declined to comment, which indirectly confirmed its authenticity, but could also have been simply a reluctance to give credence to yet another conspiracy theory. Independent researchers who did not have access to the Vatican archives tried to verify the leaks using their own methods. In 2018, a group of American geneticists requested DNA samples from several churches that owned relics, under the pretext of academic research into the history of Christian communities. Some churches agreed. The results were published in a peer-reviewed scientific journal and showed that the samples did indeed contain ancient human DNA of Middle Eastern origin. No anomalies were found, but the researchers noted that their sample was limited and they did not have access to the most significant artifacts, which are kept under strict control. Three camps have formed in the scientific community. The first consists of skeptics, who believe that all talk of genetic anomalies in relics is either the result of analytical errors or conspiracy theories. In their opinion, ancient DNA is so degraded and contaminated that any unusual sequences can be explained by artifacts of sequencing methods or contamination. The second camp consists of cautious researchers who admit that there may be something unusual in the samples, but that this requires additional verification and open publication of the data. They criticize the secrecy and call on the Vatican to open access to the samples for independent examination. The third camp consists of enthusiasts who are convinced that the leaks reveal the real picture, and that genetic analysis of the relics has indeed revealed something fundamental that the Church and the scientific establishment are hiding for ideological or political reasons. This camp is active on the internet, but has virtually no representation in academic institutions. The testimonies of insiders, despite their fragmentary nature and the impossibility of complete verification, paint a certain picture. According to this data, genetic analysis of several dozen of the most revered Christian relics was indeed carried out between 2003 and 2020. The results showed that some of the artifacts are medieval forgeries, some are authentic ancient remains, but without the possibility of establishing the identity of the deceased, and a small group of samples contain genetic features that do not fit into standard models. It is this last category that has led to the tightening of secrecy. Complete data on these samples has not been published even in closed scientific reports, and only a small circle of people has access to them. It is these samples that continue to be the subject of rumors, each new one adding details to the overall picture of what was actually discovered during the genetic analysis of the sacred artifacts. The question of DNA of unknown origin lies on the border between rigorous science and speculation.
When geneticists encounter sequences that they cannot classify, it does not necessarily mean anything extraordinary. The human genome contains billions of base pairs, and not all regions have been thoroughly studied. Moreover, databases of ancient genomes are still relatively small, especially for regions such as the Middle East in the 1st century CE. Therefore, when researchers find genetic variants that are not represented in reference databases, the first explanation is usually the simplest. It is just a rare variant that has not yet made it into our databases. However, in the case of some samples from the Vatican collection, these were not isolated rare variants, but systematic patterns. According to insiders, several samples were found to contain non-coding DNA segments with an unusual structure. Tandem repeats of atypical length located in regions that are usually highly conservative, i.e., they hardly change in the course of evolution. Conservative regions of the genome remain unchanged because any mutations in them are lethal or extremely harmful. If these areas do contain changes, this indicates either the extremely ancient origin of these changes or some special mechanism for their occurrence. One of the hypotheses discussed in a small circle of researchers from the Veritas project is related to horizontal gene transfer, a process in which genetic material is transferred not from parents to offspring, but between organisms. This is a common phenomenon in bacteria, which is how they transfer antibiotic resistance genes to each other. In higher organisms, including humans, horizontal transfer is extremely rare, but not impossible. It is known that there are sections of the human genome that were once viral genes that incorporated into the DNA of our ancestors millions of years ago. Could similar events have occurred in historical times? Theoretically, yes, but there is no direct evidence. If the unusual sequences in the relic samples turned out to be the result of recent, by evolutionary standards, horizontal transfer, it would be an extraordinary discovery, but not supernatural. Another hypothesis suggested that the unusual sequences are remnants of the genome of an extinct population of ancient humans with whom the ancestors of the Middle Eastern peoples interbred. We know that modern humans carry traces of Neanderthals and Denisovans, other species of the genus Homo that died out tens of thousands of years ago, in their genome. Perhaps there were other populations that we do not yet know about, and their genetic heritage has been preserved in separate lines, including those associated with biblical characters. This hypothesis is attractive because it does not require any exotic mechanisms. Everything can be explained by the usual processes of population mixing, but it is contradicted by the fact that the analysis showed that the unusual sequences are unlike Neanderthal, Denisovan, or any other known archaic human genomes. The third most speculative hypothesis, which was discussed only in private conversations and never included in official documents, was that the unusual genetic structures could be remnants of deliberate genetic modification. It sounds like science fiction, but proponents of this idea pointed to ancient texts, from the book of Genesis with its mention of sons of God, who entered into human daughters, to the apocryphal book of Enoch with descriptions of fallen angels, who defiled themselves with women. If these texts are interpreted not religiously, but as distorted memories of some real events, contacts with a technologically advanced civilization, or experiments of an unknown nature, then genetic anomalies could be their traces. Of course, this hypothesis was not taken seriously by most project participants, but it circulated in informal discussions and later found its way into conspiracy publications. Bioinformaticians offered a more down-to-earth explanation. Perhaps the unusual sequences are artifacts of the ancient DNA sequencing process itself. When DNA is severely degraded, the molecules are fragmented, and modern sequencing methods must assemble short-read fragments into a single sequence. This assembly process can create errors, especially if the sample is contaminated with modern DNA or DNA from other organisms, such as bacteria or fungi. Assembly algorithms can incorrectly join fragments, creating chimeric sequences that did not actually exist in the original genome. To rule out this possibility, reanalysis using alternative methods and fresh samples is required. But access to the relics for resampling was closed after the first wave of research. Another version is related to epigenetic modifications, chemical changes in DNA that do not alter the sequence of letters in the genetic code itself, but affect the functioning of genes. These modifications can be preserved in ancient samples, and, with certain analysis methods, give signals that an inexperienced researcher might mistake for unusual genetic sequences. However, experts in the field of ancient DNA who were consulted on this issue pointed out that epigenetic marks are not usually preserved in samples that are more than a few hundred years old, let alone 2 millennia. The truth probably lies in a combination of factors. Some of the anomalies are indeed artifacts of the analysis methods. Some are rare genetic variants of insufficiently studied populations, and perhaps a small portion represents something truly unusual that requires further study. But without open publication of data and access to samples by independent researchers, the question remains open. Each new leak of information adds details, but does not bring us closer to a definitive answer. Meanwhile, religious institutions that control access to relics are forced to decide how to respond to scientific discoveries that could potentially shake the foundations of the faith of millions of people. Religious institutions have always had a complicated relationship with science, especially when scientific discoveries touched on the foundations of doctrine. History remembers the trial of Galileo, the controversy surrounding Darwin's theory of evolution, and the debate about the age of the Earth. Each time, the Church had to choose: reject science, defending a literal interpretation of sacred texts, or seek a compromise, rethinking tradition in the light of new knowledge. Genetic analysis of relics has presented religious institutions with another challenge, perhaps the most serious in modern history. The Catholic Church responded to the first results of genetic research with cautious openness. In 2008, the Pontifical Academy of Sciences organized a closed conference to which leading geneticists and theologians were invited. The question discussed was how to reconcile scientific methods of studying relics with their spiritual significance. Most participants agreed that science examines the material side of artifacts, but cannot confirm or refute their sanctity. Sanctity is not a biological category, but a spiritual one. It does not depend on chemical composition or genetic sequence. The shroud may be a medieval piece of cloth, but if millions of believers find a connection with God through it, its spiritual value is not diminished. This position allowed the Church not to directly oppose science, leaving room for maneuver. However, when data began to emerge about genetic anomalies that did not fit into standard scientific models, the situation became more complicated. On the one hand, such anomalies could be perceived as confirmation of the special status of relics. If science cannot explain them, then they must be beyond the material world. On the other hand, the publication of such data would inevitably give rise to a wave of speculation, from pseudo-scientific to openly sectarian, which would undermine the authority of the Church as the guardian of true doctrine. Therefore, a strategy of maximum control of information was chosen. The Orthodox churches took a more conservative position. In 2011, the Russian Orthodox Church issued an official statement calling genetic research on the relics of saints an unacceptable intrusion into the sacred sphere. According to church hierarchs, holy relics are not just historical remains, but carriers of divine grace, and any attempt to study them using scientific methods is tantamount to blasphemy. This position was deeply rooted in Orthodox theology, which has always emphasized the mystical nature of holiness, incomprehensible to the rational mind. The Greek Orthodox Church took a similar position, denying access to relics for scientific research. Protestant denominations, traditionally skeptical of the veneration of relics,
reacted to genetic research with less tension. For most Protestants, material shrines do not play a central role in religious life. So, scientific discoveries about their origin are not perceived as a threat to faith. Moreover, some Protestant theologians saw genetic analysis of relics as confirmation of their criticism of the Catholic and Orthodox traditions of venerating shrines. If science shows that many relics are medieval forgeries, then the 16th century reformers who condemned the trade in relics and the superstitions associated with them were right. Judaism has taken an ambiguous stance on the issue of genetic research on religious artifacts. On the one hand, Jewish tradition has always valued knowledge and the study of the world as a way of understanding divine creation. On the other hand, when it came to genetic analysis of remains that may belong to biblical figures, halachic problems arose. Questions of religious law related to the treatment of human remains. Most rabbinical authorities have spoken out against extracting DNA from ancient Jewish burials, even if it could shed light on the history of the people of Israel. Islam, which does not have a tradition of venerating relics in the form that it exists in Christianity, has hardly encountered this problem. Islamic shrines are primarily places, not material objects. Nevertheless, when in 2016 the Turkish authorities allowed scientific examination of artifacts from the Topkapi Palace in Istanbul, which tradition associates with the Prophet Muhammad, it sparked protests from conservative Islamic circles. The examination showed that most of the artifacts date from a later period than tradition claimed. But this information was met with disbelief and accusations of an attempt to undermine Islamic history. Within each denomination, genetic testing of relics sparked debates between modernists and traditionalists. Modernists argued that faith should not fear the truth, and if science disproves the authenticity of certain relics, it only cleanses religion of superstition and allows it to focus on truly spiritual aspects. Traditionalists objected that material shrines are an important part of the religious experience, connecting believers to historical events and saints, and that destroying this connection could weaken faith. Who is right in this dispute is not so much a scientific question as a theological and pastoral one. A particularly acute problem has been how to communicate the results of research to believers. If scientific analysis shows that a relic venerated for centuries is a medieval forgery, should the Church publicly announce this, risking disappointment and alienation among believers? Or is it better to remain silent, allowing people to continue to draw spiritual strength from their veneration, even if it is based on historical inaccuracy? Most religious leaders have chosen a compromise, not to deny the scientific data, but also not to emphasize it, stressing that the value of a relic is determined not by its material authenticity, but by the role it plays in the spiritual life of believers. The question of how to distinguish genuine relics from fakes has faced the Church since the earliest centuries of Christianity. As early as the 2nd and 3rd centuries, the Church Fathers warned about false relics and fake artifacts. The Middle Ages became the golden age of relic trade. Demand far exceeded supply, which inevitably led to counterfeiting. The Crusaders brought thousands of authentic fragments of the cross of the Lord, nails from the Crucifixion, and pieces of the crown of thorns from the Holy Land. In the 16th century, French theologian Jean Calvin sarcastically remarked that if all the fragments of the cross stored in European churches were collected, they would make a ship, not a cross for crucifying one person. Modern science has provided tools for objectively verifying the authenticity of relics, but these tools do not always provide clear answers. Radiocarbon dating can determine the age of organic material to within a few decades, but it cannot say who the remains belong to. Genetic analysis reveals gender, origin, and sometimes family ties, but not the identity of a specific historical person. Even if the remains do belong to a man from 1st century Galilee, this does not prove that it is the Apostle Peter, and not any other Galilean of that era. One of the most reliable methods is a comprehensive approach that combines radiocarbon dating,
genetic analysis, isotope research, historical document analysis, and archaeological context. In 2014, such a comprehensive analysis was applied to the relics of Saint Nicholas the Wonder Worker, kept in the Italian city of Bari. The remains were stolen from Myra in Lycia in 1878 and have been in Italy ever since. Italian scientists were granted permission to conduct research as part of restoration work. Radiocarbon dating showed that the person lived between the 3rd and 4th centuries AD, which corresponds exactly to historical data about Saint Nicholas, who died around 343. Isotopic analysis of tooth enamel indicated a childhood spent in the Mediterranean region, which is consistent with the biography of the saint, who was born in Patara, in Asia Minor. Anthropological analysis showed that he was a man of short stature, with signs of arthritis, which explains medieval depictions of the saint as an elderly man, bent over by the weight of years. All the data came together to form a consistent picture. And the researchers stated with a high degree of certainty that the remains could indeed belong to Saint Nicholas. However, it is impossible to achieve absolute certainty without DNA from his relatives for comparison. The opposite happened with the relics of Saint Mary Magdalene from the French town of Saint-Maximin-la-Sainte-Baume. According to local tradition, Mary Magdalene arrived in Provence after Christ's resurrection, where she lived the rest of her life as a hermit and was buried in a cave. In the 13th century, her relics were solemnly found and placed in the basilica. But when genetic analysis was carried out in 2016, it turned out that the remains belonged to at least 3 different people, 2 women and 1 man. Radiocarbon dating showed that they lived in different eras, from the 5th to the 12th century. Apparently, in the Middle Ages, the remains of several people were collected together and declared to be the relics of Saint Mary Magdalene. The French Church reacted cautiously, noting that the veneration of the saint in this place has a centuries-old tradition that does not depend on the material authenticity of specific remains. Cases where several places claim to possess the relics of the same saint are particularly complex. According to various legends, the head of John the Baptist is kept in Amiens Cathedral in France, in the Umayyad Mosque in Damascus, in a Coptic monastery in Egypt, and in the Church of San Silvestro in Capite in Rome. Obviously, not all of these relics can be authentic. In 2010, a group of scientists gained access to samples from 3 of these locations. Genetic analysis showed that all 3 samples belonged to different people. Moreover, the dating indicated periods from the 1st to the 13th century. This meant that even if one of the samples could theoretically be authentic, the others were definitely fakes or misattributions. But which one is authentic, if any? Without additional data, it is impossible to answer. Medieval forgeries were not always created for selfish motives. Sometimes it was a pious practice. When a genuine relic was lost or destroyed, the Church created a replacement so that believers could continue their worship. From the point of view of medieval consciousness, it was not material authenticity that was important, but spiritual continuity. If a new relic is consecrated by prayers and revered with faith, it becomes a holy object, regardless of its physical origin. This logic is alien to modern scientific thinking, but it was absolutely natural for people in the Middle Ages. There are also cases of so-called contact relics, objects that have acquired holiness through contact with authentic relics or other sacred objects. Pieces of cloth were applied to the relics of saints, after which they themselves became relics of the second order. From a scientific point of view, such fabric has no direct connection to the saint, but for believers, it carries his grace. Genetic analysis of contact relics usually does not reveal traces of the saint's DNA, which can be interpreted as refuting their authenticity. Although, according to church logic, the absence of biological material does not negate their sacred status. The problem of falsification is complicated by the fact that some modern relics are deliberately created to deceive. In 2018, a network of traders was uncovered in Jerusalem
who sold tourists authentic stones from the Wailing Wall and pieces of wood from the Cross of Christ. Genetic analysis of the wood fragments showed that they were taken from modern European pine trees and then artificially aged using chemical methods. Buyers paid thousands of dollars for fakes, believing they were purchasing sacred artifacts. It is becoming increasingly difficult to distinguish a genuine ancient relic from a skillful modern fake as counterfeiting methods improve. Some forgers use truly ancient materials, such as fragments of medieval manuscripts or remains from old burials, and attribute a new identity to them. Scientific methods can confirm the age of the material, but not its connection to a particular saint. This makes the task of verification particularly difficult. The political dimension of genetic research on relics often remains in the shadow of scientific and religious debates. But in many cases, it determines which studies will be conducted and which will be blocked, which results will be published, and which will be classified. Relics are not only religious shrines, but also instruments of national identity, diplomatic assets, and objects of geopolitical disputes. Control over sacred artifacts means symbolic power that states and churches are not willing to relinquish. A classic example is the Shroud of Turin. This artifact does not belong to the Vatican, but to the Italian state, which has transferred it to the Catholic Church for safekeeping. Any decision to conduct scientific research requires approval, not only from the Church, but also from the state authorities. Italy considers the shroud to be part of its national cultural heritage and an important tourist asset that brings millions of euros to the city of Turin every year. The publication of results that could definitively discredit the authenticity of the artifact would damage not only religious feelings, but also the economic interests of the region. Therefore, the Italian government has always been cautious about further scientific research on the shroud. The situation is similar with the relics of Saint Mark in Venice. According to tradition, the remains of the evangelist Mark were taken from Alexandria in 928 and became the basis for the construction of the famous St. Mark's Basilica, a symbol of Venetian grandeur. St. Mark is the patron saint of Venice, and his image, a winged lion, is the city's coat of arms. Any scientific research that would cast doubt on the authenticity of the relics would affect the foundations of Venetian identity. When, in 2009, a group of international scientists requested permission for genetic analysis, they were refused without explanation. Unofficially, it became known that the decision was made not by the church but by the city authorities, fearing the political consequences of possible negative results. The Israeli-Palestinian conflict adds another layer of complexity. Many places associated with biblical events are located in disputed territories. The Church of the Holy Sepulchre in Jerusalem, the Basilica of the Nativity in Bethlehem, the Tomb of the Patriarchs in Hebron. All these shrines have not only religious but also political significance. Control over them means legitimizing land rights. If genetic research shows that modern Palestinians are direct descendants of the first-century population of Judea, this could be used in political rhetoric to justify Palestinian rights to the territory. If, on the contrary, the research shows genetic continuity between the ancient inhabitants and modern Jewish communities, this will strengthen Israel's position on the right of return. Both sides of the conflict understand the political potential of genetic data, which makes any research in this region extremely sensitive. That is why the Bethlehem project was conducted under the guise of neutral academic research, without emphasis on political aspects. Russia and Greece compete for the status of chief custodians of Orthodox shrines. After the collapse of the Soviet Union, the Russian Orthodox Church actively returned relics that had been taken out of the country or confiscated during the Soviet period. Each such return was accompanied by solemn ceremonies attended by senior government officials, emphasizing the connection between the church and the state in modern Russia. Genetic testing of the relics could confirm or refute their authenticity, but the Russian authorities and the church prefer not to take any chances. Publicly refuting the authenticity of a revered shrine
would be perceived as a humiliation of national dignity.
The Balkan countries, Serbia, Bulgaria, Greece, Macedonia, use the relics of saints as elements of national identity in the context of complex interethnic relations. The relics of Saint Sava of Serbia, Saint Clement of Ohrid, and Saint John of Rila are not just religious shrines, but symbols of national identity. Scientific research that could cast doubt on their authenticity is perceived as a political attack. When, in 2017, the Serbian authorities allowed conservation work to be carried out on the relics of Saint Sava, including minimal scientific analysis, it sparked protests from nationalist groups who claimed that Western scientists wanted to discredit Serbian shrines. Catholic countries in Latin America also see relics as a tool of cultural and religious identity. The cloak of the Virgin of Guadalupe in Mexico is not only a religious icon, but also a symbol of the Mexican nation. Millions of pilgrims visit the basilica every year, bringing in significant revenue. Any scientific research that could cast doubt on the miraculous origin of the image on the cloak will be met with resistance not only from the Church, but also from the state. International organizations such as UNESCO include many relics in their lists of World Cultural Heritage, adding another layer of bureaucracy and political interests. World Heritage status brings prestige and tourism revenue, but also imposes conservation obligations. Genetic research requiring sampling may be seen as a violation of conservation principles, even if it is scientifically safe. Who benefits from concealing the results of genetic research on relics? The list is long. Religious institutions that fear undermining faith, states that use shrines as instruments of national identity, the tourism industry that thrives on pilgrims, political forces that may lose symbolic resources in geopolitical conflicts. Each of these groups has its own reasons for keeping the full truth about relics hidden, or, at least, carefully controlled. This does not necessarily mean a malicious conspiracy. Rather, it is a coincidence of interests of various actors, each of whom acts within their own logic, but all together create an impenetrable wall of secrecy around the most sensitive discoveries. The publication of the results of genetic research on relics could have consequences far beyond academic debate. We are talking about a potential revision of fundamental religious dogmas that have been formed over 2 millennia. If it is scientifically proven that the DNA on relics associated with Christ and his entourage contains genetic anomalies that cannot be explained by modern biology, it will raise questions about the nature of divine incarnation. Christian theology asserts that Jesus was fully human and fully God. But what does that mean on a genetic level? If his DNA differed from normal human DNA, was he biologically human in the full sense? Different Christian denominations will answer this question differently, which could deepen existing divisions or even create new ones. The Catholic Church adheres to the dogma of the Immaculate Conception of the Virgin Mary and the virgin birth of Christ. From a genetic point of view, virgin birth would mean that Jesus received DNA only from his mother, without the involvement of a biological father. This phenomenon, called parthenogenesis, is extremely rare in mammals and results in the birth of a female rather than a male, as there is no Y chromosome. If genetic analysis showed the presence of a Y chromosome in the remains attributed to Christ, it would mean either the involvement of a biological father or some mechanism unknown to science. Both interpretations create theological problems. The first contradicts the dogma of virgin birth. The second requires the recognition of a miracle, which takes the event beyond scientific explanation, but also raises the question, why does a miracle leave material traces that can be studied by genetics? Orthodox theology approaches this question differently, emphasizing that the divine is by definition not subject to scientific analysis. An attempt to study the genetics of Christ is perceived as a methodological error.
the application of material methods to spiritual reality.
Therefore, for orthodoxy, the publication of any genetic data on the relics of Christ will not create a theological crisis. This data will simply be recognized as irrelevant to questions of faith. However, ordinary believers may react differently, and the Church understands this. For Protestant denominations, especially liberal ones, the results of genetic research may be taken more calmly. Many Protestant theologians have long interpreted the Gospel narratives not as a literal biography, but as theological texts that convey spiritual truths through historical events. For them, it is not the material authenticity of relics that is important, but the spiritual reality symbolized by the story of Christ. However, conservative evangelical and fundamentalist movements, which insist on the literal truth of the Bible, will face a serious challenge if genetic data contradicts the biblical narrative. The future of genetic archaeology of sacred artifacts balances between two opposing scenarios. The first is the gradual opening of access to relics for independent scientific research, the accumulation of data, the publication of results, and the formation of an objective picture of which artifacts are authentic and which are not. This is the path of transparency and scientific integrity, which, however, requires religious institutions to be willing to risk their authority and face uncomfortable truths. The second scenario is further tightening of control, classification of research, and persecution of those who try to make information public. This path preserves the status quo, but fuels conspiracy theories and deepens the divide between science and religion. Which scenario plays out depends on many factors: political will, public pressure, the position of the scientific community, and religious leaders themselves. Genetic analysis technologies continue to improve. Modern methods allow DNA to be extracted from increasingly ancient and degraded samples. 20 years ago, a relatively large piece of tissue or bone was required for analysis. But today, a few cells are sufficient. Non-invasive methods allow genetic material to be collected from the surface of artifacts without damaging them. This removes one of the main arguments against research: the need for partial destruction of relics. In a few years, it will be possible to conduct genetic analysis without any physical contact with the artifact, using remote methods of collecting biological material. Another area of development is the creation of international databases of ancient genomes. Such databases already exist, but they are fragmentary and incomplete. If a representative sample of genomes from the Middle East population from the Bronze Age to the Middle Ages can be collected, it will allow for a much more accurate interpretation of genetic data from relics. It will be possible not only to date the remains and determine their geographical origin, but also to reconstruct family ties between different artifacts and construct genealogical trees. This will turn the genetic archaeology of relics into an exact science, capable of providing unambiguous answers to questions of authenticity. Ethical consensus may also evolve. The younger generation of believers, who grew up in an era of scientific and technological progress, are less inclined to perceive scientific research on religious artifacts as blasphemy. For them, it is not so much the material authenticity of relics that is important, but rather the spiritual experience that can be gained through religious practice. If this trend continues, in a generation, religious institutions may become more open to scientific research without fear of a mass exodus of believers. Genetic experiments on relics have opened Pandora's box, from which it is impossible to put back what has already come out. The knowledge that sacred artifacts can and should be studied using scientific methods has become firmly established in the public consciousness. The question is no longer whether to conduct such research, but who will control it, what results will be made public, and how society will integrate this knowledge into the existing system of religious beliefs. History shows that attempts to suppress scientific knowledge for ideological reasons are rarely successful in the long run. The Church was unable to stop the Copernican revolution, unable to ban the theory of evolution,
and will not be able to prevent the genetic study of relics. Sooner or later, the complete data will become public knowledge, either through official publications or through leaks. The only question is whether this will happen in an organized manner, with public opinion prepared and theological reflection, or chaotically, giving rise to conspiracy theories and religious crises. The optimistic scenario assumes that religious institutions will find a way to integrate scientific discoveries into their doctrine, as has happened in the past. The Catholic Church has recognized evolution as compatible with Christian teaching, and Orthodox Christianity has found a way to coexist with modern cosmology. Similarly, genetic data about relics can be interpreted theologically without destroying the foundations of faith. The pessimistic scenario envisions a deepening conflict between science and religion, a split within denominations between those who accept scientific data and those who reject it. This could lead to a weakening of traditional churches and the growth of fundamentalist movements that reject modern science entirely. An intermediate scenario is also possible. Public attention will shift to other topics. The intensity of the debate will subside, and the question of the genetics of relics will remain the subject of highly specialized research that has little impact on the everyday religious life of most believers. In any case, one thing is already clear. The boundary between faith and knowledge, between the sacred and the profane, between the spiritual and the material, is becoming increasingly permeable. Genetic research on relics is just one of many examples of how modern technology is penetrating areas that for centuries were considered exclusively religious. Neuroscience studies religious experience, psychology explains faith, and genetics investigates holiness. Will there be any room left for the mystical and the unknowable in a world where more and more phenomena are being explained scientifically?